Hi, and welcome to your eyeball notes. These notes says special senses right here on this, but we're going to focus on the eye in this particular video. In general, you have your senses are receptors for information from the outside environment, right? We've already talked about the general senses of touch, so those include temperature, pressure, and pain, as you can see here. And then you have special senses, and they vary from just like small receptors like smell and taste to complex organ systems like your sight and your hearing, and also your ear equilibrium, which is involved in your ear. Today we're going to focus on sight. Your eyes encompass 70% of all of the receptors of your body. You have over 1 million nerve fibers in each eye. The way that we protect the eye is threefold, using accessory structures, which we'll talk about in a second, and then also, your eye is enclosed in a bony cave that's called an orbit, and also you have fat, a lot of fat actually, around your eye, and you'll see that when we dissect the cow eye. Accessory structures are eyelids, eyelashes, glands, and something called a conjunctiva. So let's start with your eyelids. Your eyelids meet at what we call a canthus, which is the edges of your eyes, the corners there. And your eyelids have eyelashes on them. You can take a second to fill this in if you'd like, and then go on with the notes. You also have oil glands in your eyes, and you can see those. Let me see if I can find my just highlighter here. These right here are called meibomian glands, and they're literally oil glands that are in your eyelids, and they help to lubricate your eye. You also have ciliary glands, and I'm going to write this out because the word cilium, you can write this, cilium equals eyelash. You can write this in your notes. And these are modified glands. These are sweat glands. And you can't see them in the picture here. They're actually between your eyelashes. And then you have a conjunctiva, which is a thin membrane that lines your eyelids on the inside, and then it connects to the surface of the eye. So it helps to keep dirt and stuff out. It's like a little sac that keeps dirt out of your inner eyeball sometimes. And it also secretes mucus, which lubricates the eye. It ends at the edge of the cornea by fusing with the corneal epithelium. So it fuses with the corneal epithelium. So remember, that's a connective tissue that is a lining, corneal epithelium. Okay. So then we talk about your lacrimal apparatus. These are your tear ducts. Just how you create tears, right? You have a gland. It's an exocrine gland because it has ducts, and the ducts are called canals. No, the canals are different ducts. They're not ducts. They drain the fluid from your eyes. So these are, let's write this, exocrine, because they have ducts. Exocrine glands. And you can see here they have a duct, and then it secretes its tears into your eye here. So how does the tears get out? Sometimes you cry and they come out of the front of your eyeball, right? Your eyes water. But there's also little canals here and inside your eye that help all of these fluids and mucus and gland secretions to drain, and it drains into your nasal cavity through this thing called the lacrimal sac. So you have a superior and an inferior lacrimal canal, and then you have a lacrimal sac. And then the duct, there's the nasolacrimal duct, which is down here. Then that's where how the fluid gets into the nasal cavity. What is the function of the nasal apparatus? Oh, what are the properties of it? First, what does it contain? Salt solution, antibodies, and lysozyme, which destroys bacteria. Its function is to clean, protect, moisten, and lubricate, of course. Irritation and also emotional upset can cause spillover into the nasal cavities, causing congestion. So right when you cry, you get all snuffly a lot. And a fascinating thing is that we don't really understand why we release tears when we have emotional upset. Other mammals do not do that. Elephants, I think, do, but I think that we'd have to look it up. Maybe look it up. What other animals cry? Okay, page 2 is your eye muscles. I'm not going to ask you to know these. We're just going to label them so that you kind of get an idea of where they are and how they work. There are 6 of them, and they move your eye up, down, sideways, sort of in and out even. I'm looking, so elevating, depressing, turning laterally, and turning medially. The nerves that activate these muscles are cranial nerves. So we can write these down. Cranial nerves. Oops, wrong thing. Cranial nerves 3, 4, and 6. So this is your oculomotor. It's kind of interesting, actually. The cranial nerve 3 is called the oculomotor nerve, and it does 4 movements. It moves the eye medially, it elevates the eye and turns it laterally, and it depresses the eye. Your 4th cranial nerve, the trochlear nerve, depresses the eye and turns it laterally, and your 7th, the abducens cranial nerve, if you remember these are the ones that come right out of your brain, moves the eye laterally. So different nerves have different particular functions. Okay, take a moment to finish filling that in, and then, so pause it and then move on with the notes. Okay, let's talk about your eyeball itself. There is, of course, you have layers of tissue around your eyeball. These are called tunics. I know. There are 3 of them. There's a fibrous tunic, which is the outside layer. It composes the sclera and the cornea. Okay, so where's our sclera? If I'm looking here, here's your cornea, right? This is what is on the front of your eye, and it turns into the sclera. It's the same thing basically, only it's a different color. It has different pigment in it. And this is the outside covering of your eye. There's a choroid layer, which is in the middle, and I'm looking for it here on your picture. Okay, here it is. And the choroid layer is, I think, going to be the vascular layer. So you can, here you have the tunics pulled away. So here's the sclera, here's the choroid. This is the very vascular layer. We'll talk about that, I think. Yeah, more specifically. And then you have the sensory tunic, which is really innervated. This has all the nerves in it. And this becomes, where does it say your retina? Yes, the retina, which houses the rods and cones, which sense light and color in your eyeball. Okay, the fibrous tunic. In the sclera, it is white. It's what we call the white of your eye. In the front, it becomes a cornea, and it's transparent. And it's transparent in order to let the light through. It repairs itself extremely easily. It also replaces itself, this is kind of interesting, every week to 2 weeks. It is the only human tissue that you can transplant, the cornea, without fear of rejecting it, so that you don't have to take like anti-rejection medicine because it is non-vascular. Okay, the middle layer, this is the very vascular layer, the choroid layer around your eyeball, and it is rich in blood. We're on page 3. It has a dark pigment to it, and that prevents the light from scattering inside your eyeball. It is modified into smooth muscle structures. So one of them is the ciliary body, and the ciliary body is the lens is attached to it, and so it is what adjusts for near and far vision. So let's write that down. So this is vision adjustment. Okay, and then the iris is the other modification. And it is the thing that dilates. It's literally, it's called a, I can't remember what it's called right now, sphincter muscle. So a circular muscle is a sphincter muscle. I know, haha, sphincter muscle, sphincter, because it's round. And your pupil is just the opening through the iris. So the iris is pigmented, it gives your eye its color, but it also is what dilates your eye for vision, okay? So it regulates the light entering, sort of like the diaphragm of a camera. Okay, the inside layer is called the sensory tunic, and this is the one that has lots of nerve endings. It also, nerves. It also contains the rods and cones, your photoreceptors, and these signals pass from your photoreceptors, going through my notes here and your book, via bipolar neurons and ganglion cells to neuron chain. We'll study this more specifically, I think, in a second. And then it runs into your optic nerve and goes to the back of your brain, your occipital lobe. Leaves toward the brain, so let's write occipital lobe. Okay, through the optic nerve. All right, here's a picture. It's at the bottom of your page, the neurons of the retina. So I'm gonna, let's see. This layer right in here, right? So here's the sclera. And then this little tiny layer is the choroid, the vascular, and then there's this layer. These yellow things here are the ganglion cells, so this is where the neurons connect to. So all this information passes down into here. So you can see on the right-hand side, you have rods, you have cones, you have cells and ganglion cells that become the axons that take the information to the occipital lobe. So, yes, take a moment to fill that in. Stay on this page because we're not done yet. Okay, the difference between rods and cones. Your rods are the most numerous. They are also more sensitive, and they are what allow basically your light vision and your peripheral vision. There is no color, though, in your rods. It's just gray. Animals have a lot more rods and almost no cones, I think, and so they're not able to really see in color like we are. We have having cones, which give us our color vision. Most of our cones are at the center of the retina, and we'll see this, I think, in a second also. Will we see this? Yeah. There's this thing called the fovea centralis, which is an area of the retina that only has cones, and this is your, like it says, your greatest visual acuity. So this is the point of your sharpest vision, your best vision. Is it in your picture? It is not in your picture yet. And then you have a blind spot, which is kind of interesting. And the reason that it's a blind spot, it's called the optic disc. And the reason it's a blind spot is because you have no photoreceptor cells there. And why is that not in our picture yet? So when light is focused directly on this optic disc, it disappears from our view. This is kind of a graph of the area of your eye. So if you go over your retina, right, that's the x-axis. So here's your nose over here, and here's your temple. Okay. So this is in the back of your eye. Here's the receptor density, which is how many receptors you have. Okay, and this line here. Do you have this picture? Yeah. You have lots of rods all across your eye, except right here. Remember, there's no. Wait, it's not even. Oh, here's the optic disc right here. So there's no receptors there. So there's a gap. And then pretty close next to that, you have the fovea centralis, where the rods go to almost to nothing, but the cones peak right here. So you have a few cones, but only cones at the fovea centralis. And this is your area of sharpest vision. You have three kinds of cones. There is one that responds to blue light, one responds to green light, and then the red cone actually responds to both green and red light. But it's the only one that responds to red light, which is why they're called red cones. If you receive impulses to more than one type of cone at the same time, that's how you end up with intermediate colors, anything besides red, green, and blue. If you get impulses for red and blue color receptors, you're going to see purple. If all three types of cones are being stimulated, like all these wavelengths of visible light, think about the visible light spectrum, right? 400 to, 660 nanometers. So this is, right, the blue down here is really low wavelengths, red. This is every color we see in the visible spectrum. If we have light of all wavelengths coming in, we're going to activate all three kinds of cones, and that's when we see white light. This is kind of cool. If you shine a red light into one eye and a green light into the other eye, you will see yellow. And what that tells us is that the mixing or the interpretation of colors occurs in your brain, not in the retina itself, not in your eyeball, but at your brain, at your occipital lobe. Okay, let's talk about the lens. The lens is the coolest part about the cow eye dissection, and if you've done it before, you might agree with me. It's like a crystal. I don't even know how to describe it. It's oval. It's not flat. It doesn't look like a contact lens. It's more like a weird oblong bead. And it's not hard, it's harder, it's the hardest part of your eyeball. It's not super hard, you can bounce it. It's held in place by these ligaments that are attached to the ciliary bodies. So remember we said the ciliary bodies, which are here, are this part of the vascular choroid, and they're muscular, and they hold these ligaments, and then they pull on them, or relax them, and that's how your eye adjusts its vision. We can also talk here about, this picture is what shows the fovea centralis right here. So this is your best line of sight, your clearest vision. And then the optic disc is the spot, and it makes more sense here why the optic disc exists, because it's where all of those nerves head into the optic nerve, and so there's sort of this blank spot right there. So this is your blind spot, the optic disc. And this is the fovea centralis, where you have all cones and no rods. Okay, fluid in your eyeball. You have two kinds. The first one is called the aqueous humor. It's called a humor. It is the water that's between your lens and your cornea. Okay, so this is just a small area. They maybe go back and look. I'm going back to look at the picture of between the lens and the cornea. So just in the front of your eye, in front of the lens, and then the iris and the conjunctiva, I think, keep that fluid in that space. It's similar to blood plasma, which means it has salts and nutrients and all those things. Actually, we haven't talked about the blood yet. So it's secreted by that choroid, which I'm looking for in the picture, which is the vascular layer, the vascular tunic of your eyeball, right? So its job, the aqueous humor, is to maintain pressure in the eye that you see. So the lens keeps its shape, and so you can see well. It also gives nutrients to the lens and the cornea. And I think it's being replaced consistently, and it's reabsorbed into your blood via that venous sinus, the canal of Schlemm, which is in your eyeball picture. Which is a canal that's in the sclera right below the cornea, so right where the clear part of your outside layer becomes the white part of the outside layer. You can see it in your picture if you don't look for it. Okay. The other kind of fluid is the vitreous humor, and this is the one that fills most of your eyeball. It's gel-like. Also similar to blood plasma, so has some salts and nutrients and antibacterial fighting things. And it is not replaced. If you damage your eye and poke it and let that fluid out, you will lose your eye. Its job is to provide a space for light to go through the lens and back to the rods and cones in your eyeball, in your retina. And so in that way it keeps the eye from collapsing. Okay, let's talk about how the sight works. Okay, how your lens sees. The light has to be focused on a point, right? And your best vision is distance vision, which means over 20 feet away. And in order to see objects that are closer, the lens has to change shape. And it does this by contracting those ciliary bodies and the suspensory ligaments. And so pulling them tighter. And so this picture here, let me make sure I'm not missing anything. This is called relative convexity of the lens. So if we're at light from a distant source, the light's coming in here, these ligaments are relaxed and your lens is flat. If you need to see closer, then these pull away and it tugs on the inside, and so creates more of a convex shape so you can see close vision. And this adjustment, that's what accommodation is called. The ability of your eye to make the lens more convex in order to see objects up close. The image that is formed by your eyeball is not how we actually see it. What we do with our brain is accommodate for what's happening from the lens bending activity. What you're seeing here in this picture is what we call a real image. So the real image here, this real image, is reversed upside down and smaller than the object. Where we have vision problems are when the lens is either too strong or too weak, or if there's some kind of structural abnormality to the shape of your eyeball. There's a section in your book on page 262 about nearsightedness, farsightedness, and astigmatisms, which are the the most common kinds of vision problems that we see. So this slide and picture, oh, we're on the last page, awesome, shows how the information gets to your brain and how it's processed. And so all of the photoreceptors here form the optic nerve, which becomes this, and then at that optic chiasma, which we saw in the sheep brain, right, they cross, but only some of them cross. And so you end up with part of the same eyeball and the same size. You get tract nerves from that, and then you get half crossover from the other eye. And then all of those nerves go to the thalamus, which is pictured here. Remember, the thalamus is in the middle of your brain, in the thalamus. And those axons here then form what they call optic radiation. So it sends it out to different portions of your occipital lobe, right? For visual interpretation. So all of your visual interpretation happens here. Another thing to note is how your eyes actually see different things, different parts, right? But because they're set forward, there's a lot of overlap of what both of your eyes see. And this is what gives us three-dimensional depth perception. It's called binocular vision. Okay, so the last thing we'll talk about is your reflexes, which is that your eye is under both somatic control and autonomic. The internal eye muscles, which are the ciliary bodies, which pull on the ligaments that adjust the lens, and also the muscles of the sphincter muscles of your pupil, are autonomic, right? And so if your eyes are suddenly exposed to a bright light, your pupils instantly constrict. That's called photopupillary reflex. And also your pupils constrict when you try to view an item really close. And that gives us more acute vision if we limit the amount of light that comes in when we're looking at something close. It gives us actually a more acute image. The somatic part are your external eye muscles, and let's see. These muscles are controlled by the cranial nerves 3, 4, and 7, which we talked about already. Convergence is that idea of we can turn our eyes toward each other to view something that's very near to us. When we perform a function like working on the computer or reading, we're using all of those muscles, both somatic and autonomic, right? We're looking at something fairly close. So our ciliary bodies are working, the muscles of our iris are working in order to converge onto the object that we're looking at. Also, our eyes are moving back and forth across the page. This is why we get eye strain, what's called eye strain, which is long periods of reading or computer use, right? So what is, of course, you probably know this, recommended is that it's important to look away and stare into your distance. Use your distant vision, which relaxes all of those intrinsic autonomic muscles of your eye. It also gives your extrinsic or somatic muscles a chance to stop moving and to just rest and look away. Okay, that's it for the eyeball.